Hello everyone, today I'm going to be going over some of the structures of the testes and epidermis, specifically with the histology portion, just because I feel like a lot of us sometimes might struggle more with understanding the different structures of the histology portion of the lab. So I just want to kind of go over the histology of the testes and epidermis mainly. And I will go over some of the actual anatomy portion of the testes, just so we kind of have a general understanding of the pathway that sperm goes through during spermogenesis. That way we can make everything else easier to understand when we go through the histology. So yeah, without further ado, I'm gonna go ahead and kind of record this way, just because I did write on a whiteboard, just because I feel like pictures help a lot. And especially with histology, noticing little tiny differences in pictures really is going to help a lot differentiate different structures. And also, I guess I can also the colors really make a difference, because when you look at everything in histology, kind of everything is stained in the same color. The only thing that helps us differentiate which structure is which is the shape of it. So I'll go ahead and I guess I kind of color coded everything, so that way you guys can kind of see little tips and tricks of how to differentiate certain structures. But yeah, without further ado, I'll go ahead and get that going. So the first portion I want to talk about is the seminiferous tubules, and basically this is going to be one of the main structures that we kind of first start spermogenesis in. So here I kind of drew a picture of the scrotum, and this is basically your testicles, and this structure here is kind of the cord that attaches to the actual testicles. So inside the actual testicles, if we actually cut open inside of it, we'll see these little tiny spaghetti strands, and these spaghetti strands will eventually come out of their capsule and go into this like bigger capsule and attach all together and kind of form what we know as the seminiferous tubules. And the seminiferous tubules are actually really important, because what they do is they actually help us produce sperm. So this is the very beginning of where we have sperm being produced, is right here inside the actual if you will, like the actual scrotum. We have a lot of like structures that are part of the scrotum, but this is the first portion where sperm actually begins to be produced. Then after that, we will actually travel kind of upwards where the epididymis is actually located. And the epididymis is also kind of like this kind of spaghetti bunch. And what the reason why the epididymis is very important is because we actually store and we actually mature cells, the sperm cells in here. And after we basically mature and store the sperm cells in here, they will go through the vas deferens. So the way that I kind of like drew this out is I also drew pictures next to it, because it actually helps me a lot. So I hope it's able to help you guys as well. But the way that I look at the seminiferous tubules is kind of like the warehouse. And in the warehouse, we kind of have where we're producing all these different things. And then after that, we have, the trucks, which basically get it loaded and prepared, just so that way it can go then through the highway, the vas deferens, and go on its merry way to wherever it needs to go. So that kind of is the same thing for the scrotum. So for the scrotum, we have the seminiferous tubules, which actually make our product, so our sperm. Then we have the epididymis, which kind of, gets it prepared, it doesn't literally box it up, but, we mature it. We get it ready so that way it can go and get sent through the vas deferens on its way through the different structures of the body. That way it can then come out the urethra, and it can come out in a product known as semen. So that's basically the general structure of the scrotum. And the epididymis, as long as the structure and histology of the actual sperm, you'll notice that it kind of has these same structures here. So right here in this main structure, we're gonna have the seminiferous tubules, which is the cross section that we're looking at right now, and up here is going to be the epididymis, and then this other portion that kind of goes downwards, which, like I said, is kind of similar here, is going to be the vas deferens. So we have the first cross section, which is the seminiferous tubule, which is made of simple columnar epithelium. And the way that I was able to differentiate some of these different structures was mainly just by the shape of it. So usually the difference between the seminiferous tubules and the epididymis, which you'll see later, is that we actually don't have a border towards the center of the seminiferous tubules. We just have kind of this blending of colors coming into one, and then after that you have these little squiggly lines within the center of the actual cell. So So in the center of the actual cell, this is what we will know as the seminiferous tubules, which will be right here in brown. Now, the spermatids, or the developing sperm cells, are actually going to be these little like linear structures. They kind of look like dots, except they're more linear, and they're kind of like in the, I guess, inner portion of the cell. And then after that, we're going to have these bigger dots, which are going to be known as spermatocytes. And those are going to be kind of, you're going to kind of see that they're kind of ordered different in different ways, but they're going to be a little bit bigger than the succenticular cells. So if we look into the microscope on a high power lens, we're going to be able to see kind of this structure right here in the center, which is going to be known as the seminiferous tubules. And then right here is going to be the little sperm. This little sperm is going to be right here. These little tiny linear structures, they kind of look like dots, except they're more stretched out, and they look a little bit more like linear. And then of course right here we're going to have the succenticular cells, and then as well as these tinier dots, which are known as the basically the spermatocytes. Or actually, I believe the other way around. Sorry, spermatocyte, which is these bigger cells, and then the succenticular cells, which are going to be these other little small cells right in between. So now that we went over the seminiferous tubules, next is actually going to be the epididymis, and this one's a little bit more easier to identify the different structures, just because you're going to have this kind of donut looking shape, and inside the donut we have what we're going to know as the epididymis. So you're going to have the same kind of general structures, and I'll go ahead and show it over on the whiteboard. That way it's a little bit easier to identify and see, but you really are going to have more of like the similar structures. So one tip that actually helped me identify the difference between the epididymis cell and the actual seminiferous tubule cell is just that it's the main portion actually has like kind of a structure to it, whereas in the seminiferous tubule we kind of saw this blending of colors going towards the inside of the cell. But right here in the epididymis we actually have this clear border, and everything's a little bit more organized. If you can kind of see in the picture, it's a little bit more organized, and you have some structures that are more defined. So you're gonna have things like the sperm, which are gonna kind of be surrounding the same portion, which is the inner portion of the cell. So if you were to look at it in the shape of like or in the form of basically a donut, the sprinkles, which is kind of like on the more inner side of the donut, would basically be the sperm, and it would be the same for the histology. And I just kind of want to bring this up just because it's like a very interesting fact that I found out about, which is that blue balls, which occurs in men, it's I don't know how, I guess I don't know how frequent it happens, but I just kind of found it funny and interesting. It's that blue balls are actually kind of known as epididymal hypertension, and what actually occurs is whenever you have an erection, it you obviously have blood flowing into the penis, and you're gonna have a bunch of blood getting built up in there, just because it's gonna make the actual tissue get kind of get erect. And what actually happens during blue balls is that if you don't have a if you have an increased amount of blood building up in the testes, specifically kind of like in the epididymis, kind of that around that region, if you have too much of a buildup of blood and you basically don't ejaculate, it can cause these pains in your your testicular region, and it actually really hurts a lot. And I just thought it was very interesting, just because when you get down to the science of it, you kind of understand what blue balls are. And this is the other portion. This is the other topic that I kind of want to bring up, and it's kind of just something very interesting. So right here I have kind of a cross section of the penis, and basically this pink portion is going to be known as the corpus cavernosum. This portion at the orange is going to be known as the corpus spongiosum, and then in the middle we have the urethra, and we have another structure on the side which I'll show right now. But basically what happens during an erection is you have all these structures getting filled up with blood, so you have blood filling up this portion, and then this portion, which is the corpus cavernosum and the corpus spongiosum. And what actually happens is these structures begin to engorge with blood. They begin to fill up, kind of like how you would a balloon, you fill up a balloon with water, it starts to, eventually build and get bigger. So it kind of happens the same thing with a penis, and it gets bigger until, eventually you have an erection. The only difference between the corpus cavernosum and the corpus spongiosum is that the corpus cavernosum will build up and engorge all the way with blood, versus the spongiosum actually won't, because the structure that surrounds the spongiosum is going to be, the urethra. So on the actual anatomy textbooks, it kind of looks something similar like this. So we have the corpus cavernosum and we have the corpus spongiosum right here side by side. So basically, what I kind of picture that would happen is, if let's say you did have both structures that engorge with blood to the max that they can, and it kind of surpasses that, you'll basically end up getting this protrusion, and basically it'll kind of pinch or block the urethra from actually being able to do a lot of things, specifically, sperm and also urine goes throughout the urethra. So if we have this pinching or this kind of a protrusion of the urethra, it'll cause us to not be able to get our waste out, like, urine, or be able to produce not produce, but I guess be able to get out sperm from our body. So I just thought this was very interesting little fact, but I really hope that the histology tips and tricks that I have on the epididymis and the testes helps out a lot. But a lot of the structures that you're gonna see it's going to be very, very similar to what the actual testicle looks like. So when you get a slide, it's going to look kind of like this, and it's going to look very similar to the, like if you know your pathway of sperm, you'll know that it'll begin here, and then it'll go up, and then it'll go out through this, vas deferens. It'll be basically the same thing for the histology. And yeah, just little shapes are the thing that really, really helped me understand the difference between different structures of the histology. So I honestly really hope it helped you guys. Thank you.